Well, good afternoon, everybody, and welcome to our webinar fourth webinar of our virtual masterclass series this year on ensuring accurate classification of BRCA1, BRCA2, and other HRR gene variants. Next slide, please. So my usual duties relating to housekeeping. So we have automatically muted your microphones to avoid any background noise. We are recording this webinar, so you will be able to watch it again at your leisure after this live webinar. We typically tend to try and share this between 2 or 3 weeks after the webinar has taken place, but we'll notify all users and all registered participants as soon as it is available. We actively encourage you to use the Q&A function to submit your questions. This is largely geared around education associated with this topic. So please do use the Q&A function to submit your questions. We will have a dedicated section on Q&A at the end of the webinar anyway. And anything we don't pick up in that session, we will then pick up in the summary report that will be published shortly, I think in early January or February, related to this actual run itself. So make sure you use the Q&A. One key thing associated with that, please do define what case you are asking a question for. So if it's very specific to one of the cases 1 to 6, preface your question with a case number so we know and we can easily reference it quickly. Otherwise, it will assume it's a general question related to the entire webinar itself. Next slide, please. So your feedback has helped us hugely over the past, so it's very important to us to make sure we get your feedback off these webinars. We really try hard to make sure we provide you with high quality training. So you will receive an email after the masterclass with a link to a short feedback survey. It should take no more than 2 minutes to complete, but we actively encourage you, please, to keep feeding back to us what you'd like us to do, how you'd like us to improve, whether you found the webinar helpful for your educational needs and requirements. Next slide, please. So we have 3 fantastic speakers. As always, we have some really, the knowledge that we have behind the delivery of this webinar is fantastic. So we've got Professor Kathleen Klaas from Ghent, Dr. Arjen Medensenkamp from Nijmegen in the Netherlands, and Miranda Durkee from Sheffield in the UK. All of who've got very extensive experience in the classification of these variants. They're going to take a couple of cases each, and then obviously we'll come back to the Q&A at the end of it after the session. Next slide, please. So our disclaimer, just to clarify that this is a non-promotional webinar organised by AstraZeneca and Merck Sharp and Dohme. Next slide, please. So I'm going to hand over to my co-chair, Dr. Sandy Deans from GenQA, who's going to give you the background behind this run itself, and then we'll lead on to the speakers straight after that. Over to you, Sandy.
Thank you, Simon. So welcome everyone to the webinar, the 4th webinar this year. So we are going to summarise the EQA findings from the latest external quality assessment run. So this is an overview of the participation and of the classification of these 6 variants. So next slide, please. So for the first time, we've launched the online classification EQA through the new platform that's been developed through GenQA. So it's the Genie platform, and really it's just extending the GTAC system that we were using before, but to formalise it more into an individual education element. And there's many other modules there for you. So it's generated in the same way. It can be accessed through the GenQA website. It's available globally all year. These are the static variant EQAs that we run for BRCA and HRR genes that are sponsored and supported through AstraZeneca and MSD. So we've got our website link there, and hopefully you found it an easier system to use than previous versions. But the fundamental is the same, and we can deliver this on for individuals rather than laboratory participation. So next slide, please. The Run 11 that we're discussing today was delivered during the calendar month of October this year. So we had 408 registrants from 49 different countries. And I think the benefit of it being online and available whenever you want to use it demonstrates that we've got really good coverage across the globe with 49 different countries. And here's just an idea of those countries where we had 10 or more registrants. However, we did see a drop in those actual individuals who participated and completed the Run 11. So really, I think our ask to you is if you do register, and we always have a top limit of number of people who can be accepted, is we really would encourage you to actually participate, because the feedback and the general learning that you obtain really is quite important. And I think you would feel gain a lot from actually participating. So here you can see again that we have 5 or more participants from these countries here. No surprises really that the UK is most predominant, as this is a promotion that we do extensively through the labs in the UK to participate. So these are individuals participating. So next slide, please. We also have the benefit through these modules to actually find out what people are actually doing and how they go about doing the actual classification themselves. And one way about doing this is actually surveying and finding out which guidelines are in use. As always, we see there is different approaches being used. Here you can see that 26%, or a quarter of the participants, are using the ACMG 2015 guidelines only. You then have 9% who are using these guidelines, but also using the UK ACGS 2020 guidelines. Then we have, and I think this is increasing from other EQA runs, that we have 43% who are now actually using both of these guidelines, but also adding in the CANVIG UK 2020 guidance. And that probably reflects the high number of UK individuals who are participating. And then we have a further 11% who are using Enigma guidelines only, and then another 11% who are using a smattering of different guidelines that are out there, some of which are also in-house guidance. So it's very difficult to define exactly how they have come about for their classification. Next slide, please. So this is a breakdown of the background of individuals who are assessing these variants. We know that it's an individual activity, it's not a laboratory-focused activity, and we have individuals with lots of different backgrounds who are actually classifying variants for a range of different clinical cases. So by breaking down by specialty or department, you can see here that 63% are sitting within genetics or genomics laboratories or clinical units. Then we've followed with 15% who are based in oncology, 14% who class themselves as pathology-based, and then we have a further 8% who are other different specialties. Then we wanted to find out who were the types of individuals who were doing these classifications. And here again, you can see that probably linked to the genetics element of the departments, that 46% were actually from experienced scientists, that these are self-defined experienced scientists. The next highest area is a 29% who are other, and I think we need to maybe find out and break this down to find out what other actually means, because I think we need to target this education element specifically to the needs of our participants. Then we have 11% who are recently qualified scientists. So again, it's very much into a registered clinical scientist element of the workforce. Then we have both 4 or 5% for the remaining, which are medics or pathologists or students. So I think it's really great to see that we have a wide spread of different backgrounds, not just in specialties and departments, but the different roles that people are performing within the workforce. So thank you for submitting that data. Next slide, please. So for the Run 11, we provided 6 different variants. You can see here, as always, we like to give a full range of different classifications. So for variant 5, we were down at the benign, likely benign area. And for variant 2, 3 and 4, we were up more at the pathogenic. Now you'll see here that 2 of these variants have been starred, and this is really dependent on the guidelines used for classification. And I think this is why these masterclasses are so important, that we can actually understand how the variant can be classified differently depending on which guidelines are actually applied. So we're going to go into more depth in the webinar as to what guidelines were applied and the impact of that classification. So next slide, please. So this is the number of participants with the reported expected classification. So great news is we can see for this run, we had 17% of participants who obtained the expected classifications for all 6 variants. And it's the first time we've had such a high proportion of participants actually getting the expected results. So I think that's pure demonstration that is ongoing assessment through EQA and examples and worked examples and masterclasses really do help standardise classification. Then we also had a large proportion of participants obtaining 5 expected classifications, which again is fantastic, and then closely followed by 4 variants. So I think we can see here that there's definitely improvement in standardisation. So next slide, please. So I'm now going to hand over to Kathleen, my colleague, to give an in-depth review of the classification applied to variant 1.
Thank you, Sandy.
Thank you.
So next slide, please. The first variant is a variant in ATM. It looks like it leads to a change in amino acids. So it's a change 902G2A, but actually this is the first nucleotide of exon 8. It is located in a functional domain, meaning the armadillo ARM type fault domain. Next slide, please. What evidence is available online and in the literature? So it is the variant is reported in the GNOMAT non-cancer females, and it's present there in about 0.005% of the people present there. In LOVD, it is also reported, and there is also a prevalence, a frequency available from the Bridges Consortium, as you can see here, and it's a bit higher than what is reported in the GNOMAT non-cancer females. In the GLINVAR, it's also reported, it gets there 2 stars because all participants agree on the classification, meaning they all classified it as a variant of unknown clinical significance. There are also in silico predictions available like REVEL, Mutation Taster, Polyphen, SIFT, and also Splice AI, which is relevant because it's the first nucleotide of an exon. And as you can see, Mutation Taster, Polyphen, and SIFT all indicate that it could be deleterious. Next slide, please. This is about the evidence that we have been discussing amongst us and which evidence we would apply. And we agreed that we could apply BP7 from observed at a supporting level. As you may know, BP7 is normally applied for a synonymous silent variant for which splicing prediction algorithms predict no impact. However, the ATM VSEP guidelines interpret BP7. You can see it in a different way, and you can use it if there is also for other not only silent variants, but also other variants that may lead to an amino acid change. If there is a lack of an aberrant RNA defect, and it's the weight you attribute to it can be dependent on the assay you applied. So we used in the literature, there is one publication who have investigated this variant using a mini gene assay, and they observed actually that from the mutant allele, there is still about 65% of full-length transcript transcribed. However, they observed some protein truncating transcripts lacking exon 7 and lacking exon 7 and 8 at a lower prevalence. So we can really see that people applied PVS1 for that. However, the reason why we felt this was more BP7, it is because we are aware that some labs have done some cDNA analysis and did in-patient material and did not observe the aberrant splicing due to this variant. But we also, therefore, we could also perfectly live with the people who applied PVS1 observed at the supporting level. So the PVS1 is normally applied for a null variant in a gene where loss of function is known mechanism of disease. And the paper from Bueno Martinez et al. Indicated themselves that they would apply PVS1 as an observed splice defect. But if you apply BP7 or PVS1, you end up in a class 3, a variant of unknown significance. Next slide, please. So, and this was also the outcome from the large majority of the people who have classified the variant. As you can see here, there were a few that classified it as likely pathogenic, which is of course not advised. And on the next slide, we can see what arguments have been applied. So the PVS1 observed was applied by 35 participants and the PP7 only by 1 participant who indicated that cDNA analysis had been done. What we saw is that several people applied PM2 at different levels, so moderate or supporting. However, PM2 in the guidelines from CANVIC, but also VSAP, it's now advised to use PM2 only at the supporting level. You should note what is the cutoff. This can be gene dependent, and for the ATM, the cutoff is determined at less than 0.001% in all subpopulations. And if we check in the GNOMAD non-cancer females, you can see it was 0.005%, which is higher than the cutoff that is allowed. Many people also applied PM5, and PM5 is about a missense change at an amino acid residue where different missense change has determined to be pathogenic. When I checked and I used the CANVAR UK database, which variants have been reported in that amino acid, you can see the list here. None of them were considered as pathogenic. And on top of that, the guidelines for ATM state that you should not use this argument. So because there are many amino acids that can be pathogenic or benign, even if they are at the same codon, and the bioinformatic tools are not reliable to predict that adequately. So you can only use PM5 at the supporting level for truncating variants in the ATM gene. And then several applicants also applied the PP3 criterion. So this means these are the in silico prediction tools, and we have seen on the first slide that there were some variable outcome. It is advised for amino acid changes to apply REVEL, and you can only use PP3 at the supporting level if the value obtained is higher than 0.733, which is of course not the case because it's 0.418. Or if you realize that it's the first nucleotide of an exon, you should also look at the splice predictions, and the value obtained was 0.04, which is lower than what could be applied for PP3 and too high too high for BP4. So in that sense, it's also, cannot be applied. And I think this was the last thing I want to discuss. So I would like to hand over the word to Miranda to discuss the second variant.
Thank you very much, Kathleen. So hi, everybody. Yeah, I'm going to go through variants 2 and 3. So next slide, please. So starting with variant 2, this is a BRCA1 splice variant, and it's a non-canonical splice variant, so it's affecting the +6 position. So it's C.4357 +6 T to C, and it's a putative splice variant in intron 13. Next slide, please. These are the kind of common databases that and resources we would always recommend checking. So it is absent from NOMAD. It was present on ClinVar. It only had 1 star, but there are 7 different submissions, 4 at pathogenic and 3 at VUS. It is present on BRCA Exchange, but it had not yet been reviewed, so given a classification on there. Again, it was present on LOVD with 3 entries, 2 being pathogenic and 1 unclassified. So 1 unclassified was from the UK Variant Sharing Initiative, and then it was reported twice in that Rebecca et al. 2018 paper, which was a kind of good review globally of different BRCA variants. So very interestingly for this variant, the splice AI score, as Kathleen's just showed, the cutoff is below that 0.1 that would be recommended to kind of apply PP3. So in actual fact, it would score a BP4 for this variant. Also, if you look at some of the other splice tools like MaxEnt Scan, Splice Site Finder, etc., again, some of them show a very small difference in their scores, but not really anything very significant. But the next stage was obviously that you would go on to kind of search the literature and to see if any other cases have been reported in published papers. So next slide, please. And then this slide kind of reviews all the evidence, and we have classified this variant according to both CAMVIG guidelines and Enigma guidelines. So I'll start with the CAMVIG guidelines. Because this variant is absent from controls, and at this stage when we were doing the classification, we had NOMAD version 2.1 and version 3.1. So it was absent from both those non-cancer groups. There is now a new NOMAD 4 out, and so I will come to a slide in that in a few minutes. We could also then find from our literature searching that there were 3 published papers that had all done some RNA studies, and 2 of them had some quantification on it as well. So we had Gaither et al. 1995, Smith et al. In 2019, and Way et al. At 2020. And 2 of those papers showed that you had about 85% of the transcript showing it was out of frame, and the rest was wild type. So using the CAMVIG guidelines, because there's more than one assay and they're kind of, so it's reproducible, they've all shown abnormal transcripts, but there is definitely some evidence of leakiness. So that means some wild type transcript is produced. So it's incomplete splicing. We would then downgrade that PS3 to moderate. Then really the kind of key piece of information that differs between the CAMVIG and the Enigma guidelines is the case data. So in this case, we can use our CAMVIG UK data and look at the prevalence within the kind of diagnostic population and compare it to the UK Biobank population data to get a very significant p-value and odds ratio. I'm going to come to that in the next slide, so I won't go through that in detail now. So moving on to the Enigma guidelines, the same kind of evidence in terms of PM2, but as per the kind of ClinGen recommendation from a few years ago, that is used at maximum strength of supporting being absent from NOMAD. And Enigma have also adopted the recommendations from the Walker et al. 2023 splicing SVI group, where the RNA functional evidence is now incorporated under PVS1 RNA. And so for the exact same papers, again, there's a kind of lookup table, and because of you get between 80 and 90% of outer frame, so kind of leakiness, they downgrade that from very strong to strong. And Enigma set a cutoff actually of 80. If any transcripts below 80%, you wouldn't use PVS1 at all for that. So you can see that these differences in the evidence used means that the CAMVIG classification is a class 5 pathogenic, whereas the Enigma classification is class 3 VUS. Next slide, please. So this is a summary of the participants' classifications. So you can see a large number of participants got the expected classification shown in green, either based on the class 3 using Enigma or class 5 using CAMVIG. There's a group in the middle that have kind of, still called it lightly pathogenic, but maybe weren't adhering kind of strictly to either set of guidelines. And then I've just highlighted a few that have kind of incorrectly classified the variant. Next slide, please. So just to go into a little bit more detail in terms of the PS4 and the difference between the CAMVIG and the Enigma guidelines. So for CAMVIG, we are very lucky because we have all of the NHS diagnostic labs submit data to our national kind of disease registration service, and that data is kind of put shared back and put on the CAMVAR UK website. So this is the big kind of picture is a screenshot from that website. So you see the third tab along so it shows case control evidence. It shows that it's absent in NOMAD, and we've got the UK Biobank data. And again, because it's a BRCA gene, so it's mainly causing breast and ovarian cancer in females, we would select the UK Biobank female population to make it as closely matched to our kind of diagnostic testing. So you can see that we've got 2 alleles detected in nearly 24,000 cases on UK Biobank. And then on the right shows the case frequency tested in the diagnostic labs. So these will all be people with breast, ovarian cancer, and potentially some prostate cancers in there. And you can see we've got 19 patients in 62,000. So then at the bottom on the left-hand side where it says the observed contingency table, if you feed this into a Fisher's exact table, those quantities, on the right-hand side, it shows you a highly significant p-value. It also gives you an odds ratio, which again, I've used a different calculator, a little bit clearer, to show you the odds ratio on the far right-hand side. Sorry, I can't use the... I haven't got a pointer with the slides being shared centrally, but you've got an odds ratio of 36.6, so it's highly significant. So using these guidelines, we would then apply PS4 at very strong. Okay, next slide, please. Oh, sorry, apologies. Can we go back one? So I just wanted to then say how many participants use PS4. So 114 out of the 235 use PS4, 29 applied it at very strong, 62 at strong, and 15 at moderate and supporting. So we can see from this that there's definitely potentially some education kind of needed in terms of how we use this case control data. Next slide, please. Thank you. So this is now the splicing data. So again, this is a screenshot from the CAMVAR database, and you can see on the 5th tab along, there is this functional splicing analysis tab. And this kind of highlights some of the key assays that we've already mentioned, like the Gaither and the Wyatt et al. So for splicing data, 57 participants used this within PVS1 at variable strength. So 26 used it at very strong, 28 at strong, 1 at moderate, and 2 at supporting. Whereas 108 participants used that same splicing evidence within PS3, 10 at very strong, 73 at strong, 9 at moderate, and 16 at supporting. I've highlighted the very strong ones in red because we know that there is leakiness from 2 of those assays. So I think I would caution away from using that at that strength. It is probably better to downgrade that evidence. We are just in the process of updating kind of UK guidelines and moving towards that Walker et al. For splicing RNA assays to move to PVS1. So hopefully in future rounds, this will be more consistent, and we will all be using the splicing data under PVS1, but this is how it is at the moment. And then for PM2, 208 participants used it, 1 at very strong, 4 at strong, 92 at moderate, and 111 at supporting. And again, just to highlight those strengths of very strong and strong, which are kind of not allowed in any guidelines, and are really overweighting that kind of rarity and controls evidence. Next slide, please. So moving on to classification of variant 3. Next slide, please. So this was a copy number variant, and it is a deletion of exons 14 to 16. That's actually an in-frame deletion that's affecting the DNA binding domain. Next slide, please. So these are the databases and resources that we have reviewed for this. Are fairly standard to the a small variants that we would normally check, but in this case we've also checked DGV, which is database of genomic variation, which is mainly CMV data from arrays. So kind of a bit of a caveat there, but because this is a fairly large CMV, it should be kind of picked up by those SNP array studies. I have a massive caveat to add to this slide, as that these types of variants can be very hard to look up on these different databases. You can see that the HGVS nomenclature that we've used is using kind of the breakpoints are within between intron 13 and another breakpoint within intron 16. Other people might use HGVS saying the first base 7008 minus question mark or plus question mark. If people are using MLPA, they sometimes use the cDNA positions of the MLPA probes. If people are using assays that have actually looked at the breakpoints, or they've used WGS, then you will have those exact breakpoints in there. And then, of course, some people just use the words exon 14 to 16 deletion. So these types of variants are very, very hard to look up on these databases. So it may be that there are some there, but with alternative nomenclature. Next slide, please. So now the classification, and again, broken down to the CAMVIG guidelines and Enigma guidelines. So for the PVS1, we would use in CAMVIG the Teun et al. PVS1 decision tree. As this is an in-frame deletion, but affecting a critical functional domain, it's upgraded from moderate to strong. In the UK, as it's absent from NOMAD, and I've just recently checked the new NOMAD SVs and CMVs, so I'm going to come on to these on the next slide, and that database of genomic variation, then we would allow use of PM2 at supporting. And then the PS4 is the case data. So as I've kind of highlighted on the previous slide, because of that problem of finding the variant on all these databases, we can't really do a case control study. We couldn't get that information from how many cases were studied using WGS from the UK Biobank. But CAMVIG guidelines do allow the use of case counting. So we know that there are 7 cases from the national data collected, diagnostic data collected, and in the Rebecca et al. 2018 publication, there are 38 cases. So it's, you know, a very well reported variant. And so the CAMVIG guidelines allow you, if you've got 10 cases in high quality, kind of well recognized families and published in the literature. So this is definitely not ClinVar data. We do not know the phenotype of a lot of the ClinVar data, but when you've got a good publication like Rebecca, or you've got the UK diagnostic data like this would be sufficient to apply at PS4 moderate. And then we also have applied PP4 at very strong for using the Parsons et al. Multifactorial data. And again, I'm going to come on to explain that a bit more. So applicable evidence using the Enigma guidelines, again, similar evidence, but kind of slightly differently used. So using the Enigma PVS1 decision trees, which are incredible and very, very detailed, and, you know, really useful information on there. And because of that being such a critical known domain, the DNA binding domain, Enigma have upgraded this to very strong. And again, then they use the Parser multifactorial data, but Enigma do not allow the use of PM2 or PS4 for these types of variants. So although there are some differences in both classification systems, this would end up as class 5 pathogenic. Next slide, please. So this is a summary of how participants classified the variant. So the majority of participants did get class 5 as expected. That's great. Still a significant proportion did get it as lightly pathogenic, but obviously it really wouldn't have clinical implications. And then I've highlighted the ones where the classification potentially would affect clinical management. Next slide, please. So looking at the PVS1, first of all, as I said, the Enigma upgrade that to very strong, but Canvig would use that as strong. So in 189 participants used it at used PVS1, 117 at very strong, 60 at strong, and 12 at moderate, I presume, because it is in frame. And this variant, we like to do this sometimes. We previously included it in one of these earlier same GTEC runs, and it was an HRR run 6, and we previously classified it as class 4 lightly pathogenic using the Canvig guidelines at that time. So I don't know whether that was why a large number of participants went for the lightly pathogenic, kind of using the same classification as before. But this was actually classified before the multifactorial data was incorporated into the Canvig guidelines. So just looking at that multifactorial data in a bit more detail, this is a screenshot from the Parsons et al. In supplementary table 1, and they look at the likelihood ratio, and it was split down by segregation, pathology, co-occurrence, family history, and case control, and then into a combined likelihood ratio, or an odds for causality, which you can see is a whopping, it's over 50,000 likelihood ratio. And then the screenshot below shows from the Canvig guidelines how you convert that likelihood ratio into evidence points and then into a strength. And you can see the cutoff for this is 350. So this variant is way in excess, there's a huge amount of evidence for this variant, and therefore we could apply it at very strong. Only 27 participants actually used PP4. The majority of them, 23, used it at very strong, 1 at moderate, and 3 at supporting. So it was generally well used when it was used, but it was massively underused by participants. And there's a huge wealth of data kind of in these publications, such as Parsons et al. Next slide, please. And then I thought I would use this opportunity to kind of introduce the new Nomad 4, version 4. This was released on the 1st of November. It is now 5 times bigger than both the versions 2 and versions 3 put together. It includes, I think, over 400,000 exomes from the UK Biobank, and has a total of about nearly 731 exomes and over 76,000 genomes. So it's a huge data set. And quite importantly, for variants such as this, it now has, under the SV or structural variant track, more than 1.2 million high quality structural variants. Another thing that's new in this release is they now have a special data set for CMVs. And these are only for rare, high-quality CMVs that include 3 or more exons, and there's nearly 67,000 of those. So again, another really useful resource for you to look at population data for this. And as I said, Canvig allows the use of PM2 supporting the CMVs. And this is a screenshot from the SV database. It wasn't in the CMV database. I checked that as well to show that there are no SVs that include all of those exons 14 to 16. Next slide. I think that was it for me, and I'm now going to pass on to Ian.
Thank you. Next slide, please. I will discuss this missense variant in BRCA2. It is a isoleucine 2627 to phenylalanine, and it's a missense variant in the DNA binding domain of BRCA2. Next slide, please. It is absent in the Nomad database and present in the BRCA exchange as pathogenic. ClinVar reports it also 222 times as a pathogenic variant, and once as a likely pathogenic variant, once as a VUS, and even 1 time as a benign variant. Also present in LOED, and in silico predictions are given below. Revel is high. Base del is indicated now also because base del is part of the Enigma guidelines. Mutation, taster, polyphen, etc. all have been indicated here. Next slide, please. So here again, we try to classify this variant according to both CanVIC guidelines and Enigma guidelines. For the CanVIC guidelines, PS3 is applied at strong level based on several functional studies, and that same applies also to the Enigma guidelines, of course. PM2 is applied at moderate level or supporting level, relative whether you use CanVIC or Enigma, as discussed previously. And here again, the most important code is PP4, very strong, as there is a combined likelihood score for this variant of a score of 1,000 to 1. And like in the previous variant, that causes an application of the score at a very strong level. Then we go for the UK variant. You can apply for the UK guidelines. You can use PS4 moderate, as the variant is reported 15 times in HCMD and 25 times in ClinVar, and also in LOVD. So it is case counting here, and case counting is allowed according to the CanVIC guidelines, but cannot be applied according to the Enigma guidelines. So PS4 is not applicable to the Enigma guidelines. PP3 again is also different because, and that is the odd situation that people use different prediction tools. The Revel score is over 0.7, 0.795 to be exact. So that is why PP3 can be applied according to CanVIC, but the Enigma guidelines use base del, and that is too low. I'll come back to that later. Anyway, especially because of the data from the likelihood score, PP4 is the major code, and both ways together with the functional data leads to a prediction of a class 5 variant. Next slide, please. And the vast majority of you also came to a class 5 classification. A small group ended up at likely pathogenic, and 2 participants classified as variant as US. Next slide, please. If we look at the differences, like I said, PP3 is not applicable for when you use the Enigma guidelines, as the base del score is below 0.30. And PS4 mod is not allowed to the Enigma guidelines. And that is based, as it is reported in many patients, but as breast cancer is a very common, this may be due to a reporting bias. You have to have a proper controls data set to apply PS4. PM1 was applied 69 times, and that cannot be applied at all using the Enigma guidelines. That is part of the domain in which the variant is. It is incorporated in the domain. It is part of the CanVIC guidelines, but it cannot be used when functional data are being used for PS3. There is a, as per the main CanVIC guidelines, it is seen as double counting. So because the variant is functional, it is likely that the variant is an important domain, and therefore PM1 cannot be applied in either of the 2 guidelines. PM5 has been applied 57 times. And PM5 is a code that can be used as another amino acid change at that position is classified as pathogenic. It cannot be applied according to Enigma at all, as it overlaps with the PP3 code. The variant is already in a domain, and that you should not be counted more than once according to that guideline. For CanVIC UK, it principally can be used. However, if you look at the other variant that is indicated there, and that is the isoleucine to asparagine, and if you look closely, that variant is called likely pathogenic based on the fact that the variant here under consideration is pathogenic. So using it here, it would be a circular elimination, and you can't do that, of course. And therefore, you cannot apply PM5, also not when following CanVIC UK guidelines. Next slide, please. Then again, then we get at the PP4 or PP5 according to the previous CANVIC guidelines, and that is the ENIGMA data from Easton et al. That published a likelihood ratio of more than 1,000, and that gives you a PP4 at a very strong level. Some participants use the PP5 code and only referring that ENIGMA specified this variant as a class 5. And this option has been removed from the CANVIC UK guidelines. But of course, you can use the available information why ENIGMA ended up at that classification, and that is the PP4 data for likelihood calculation on the basis of segregation. Okay, thank you. The next slide, please. So now we go to the next variant, variant 5. Next slide. And this is a BRCA2 variant, and it's a synonymous variant inside the DNA binding domain. Next slide. The variant is present in the NOMA database in 2 females. It is also present in the BRCA exchange database as a likely benign variant, and ClinVar varies between benign, likely benign, and VUS. It's also present in LOVD, and Splice AI, which is now the only in silico tool that can be used here, indicates that there is no expected splicing for this variant. Next slide, please. Here, again, we looked at the CANVIC guidelines and the ENIGMA guidelines. The BP5 moderate can be applied according to both guidelines, again, based on the multifactorial likelihood analysis. Well, this one is a more difficult one, and I can come back to that later in the discussion. For the CANVIC guidelines, you can apply BP7 as it is a synonymous variant, which is not conserved at all, and it does not predictably cause splicing, and BS3 moderate level. And BS3 moderate is for a well-established functional study showing no damaging effect. And this variant has been reported in 2 studies in literature. The Sunset all reported a partial exon splicing, and Weinstein reported that you only get a normal transcript. In the report in the Sunset all, it only said a data not shown partial exon 24 skipping, but I will come back to that later in the next slide. ENIGMA guidelines uses slightly different codes. Of course, also the BP4 is here still applicable as it is a synonymous variant within a key functional domain. That is important in ENIGMA. So because it is in the DNA binding domain, you can apply BP4 as Splice AI is 0. The next you can, according to the latest rules, you can use here BP7 for RNA work, and then you get an upgrade to strong as it is a synonymous variant, again, according to the same 2 studies. Although Sans et al. Published a partial exon skipping, I got back to the authors of this paper, and the next slide you will see that this partial exon skipping is very, very limited. Then again, we end up at BP5 moderate, just as for the CANVIC guidelines, based on the multifactorial likelihood analysis. Next slide, please. The vast majority of participants ended up at likely benign or benign, and a handful of people classified this variant as a VUS. Next slide, please. Well, let's come back to the BS3. That is applied only according to the CANVIC UK guidelines and BP7 for the ENIGMA guidelines. And it is applied at strong level if you take into account that it was only a normal transcript. However, there was one publication that showed a partial exon skipping without providing any further evidence. And then it is very hard how to apply this code. We had communication with the authors, and they provided us this picture indicating here the below the vague band, and which is next previous slide, please. Yes, the arrow points to the vague band, indicating a defect less than 10%. And because it is less than 10%, according to the ENIGMA guidelines, you can apply BP7 at a strong level. So that is very helpful. Of course, BP7 moderate and strong was accepted here because this information was absolutely not readily available. And then there is an even more difficult point that following the Enigma guidelines, if you do not take into account the splicing data, you start with a BP4 and BP7, as it is a synonymous variant, not expected to be causing any splicing. However, as we do have RNA data, BP7 can be upgraded to strong, just like I did on this slide. BP4 is then still retained. However, if you follow the CANVIC UK guidelines, BP4 cannot be used in combination with the functional BS3 data. So here again, there's a difference between both guidelines, which makes it, of course, very confusing. Next slide, please. Well, we get to another difficult point about BP5 or BP6, according to the previous CANVIC guideline. We go back to the Lee et al. Paper. In the Lee et al. Paper, and this paper is very hard to find if you just look PubMed. This paper gives in the online table S4 a LOT score, an LLR score of -1.25, based on 12 probings. If you want to calculate the likelihood score, you have to calculate back. So you have to use the 10th power of this LOT score, and then you get a score of 0.055. However, it is not always clear whether you have to use a natural log or a 10th log, as we have done here. The CANVIC UK has also converted this score into a likelihood score, but used the natural log, the E-log, and came up with a score of 0.284. So if you would like to use this likelihood score and try to translate it back into a code strength, you end up at different levels. For 0.055, you will end up at moderate. You will end up at a moderate level, and 284 only at the supporting level. But however, like I said, we also here we get back to the raw data, and it is a true. The 0.055 is the true score to be used here for this paper. Of course, BP6 on the level of a reputable source should not be used. It has also been removed from both guidelines. Of course, you can use the likelihood scores based on a multifactorial likelihood analysis, but you should use that under the BP5 code from now on. So that is very difficult. This paper is very hard to get by, but the data from the Lee et al. Paper has now also been published at the CANVIC UK website, and also it is also already present on the ClinGen repository. However, it is not yet visible on ClinVar. Next slide, please. And I would like to hand over now back to Kathleen.
Thank you, Arjen. I will discuss the last variant. It's a variant in PAL-B2, and it looks like a truncating variant because it's out-of-frame deletion of one nucleotide at position 3556. It's a frame shift, and it's not located well in a functional domain. Next slide, please. The variant is absent from all the databases we have consulted, like GNOMAD, ClinVar, and LOVD. Next slide, please. What are the criteria we have applied? And for this, we have used the ACMG-AMP variant interpretation guidelines that have been published earlier this year. So we applied the PVS1 score at a supporting level. So PVS1 is for null variants in genes with established loss of function as a disease mechanism, which is clearly the case. But there is something special going on with this variant. So the most C-terminal protein truncating variant known to be pathogenic was published by Reed et al. Back in 2007. And that's a nonsense variant at position 1183, where a tyrosine is replaced by a stop codon. This means that the last 4 nucleotides of PALB2 seem to be still very critical for the PALB2 function, or at least, maybe not all, but at least some of them. And this is also supported by structural considerations that were published by Oliver et al. In 2009. What happens now when this deletion takes place at position 3556? Well, we get actually like a longer amino acids chain. So at the end of the protein, and you can see it here in red, and normally the last 4 nucleotides, the four last amino acids are YHYS. And when you have the deletion of this nucleotide, the amino acid chain is prolonged, and the last serine is replaced by histidine, and then you get the last 3 other amino acids that are introduced before you get a stop codon. And I will come back to that, why we have to downgrade it, then the evidence from moderate to supporting. It's in the next slide, but I just want to mention that the variant is also not present in GNOMAD, so we used also PM2 at the supporting level as now advised by the guidelines. And we have 2 supporting arguments, so this we end up in a class 3, a variant of unknown significance. Next slide, please. So in the guidelines for PALB2, there is a nice overview for the PVS1 decision tree. So if you have a nonsense or frameshift variant that is predicted, that is not predicted to undergo NMD, there are some options. So you can either have a truncating something that's a variant that leads to a truncation upstream of histidine 1184, and then you can apply PVS1. However, if the role of the truncated region in a protein is unknown, so if you have a nonsense variant that introduces a stop codon downstream of tyrosine 1183, then it should be moderate. And here we go even further. We have in the last line, you see that's a frameshift that's starting downstream of tyrosine 1183, and we have an alternative C terminal, and this leads then you should downgrade from moderate to supporting, so that's why we applied PVS1 supporting. What were the results that we can see in the next slide? Of the classifications. A large number of people classified it as uncertain clinical significance. However, there were a significant number of participants who classified the variants as likely pathogenic or even pathogenic, which of course has a major clinical impact, because this means that predictive testing will be offered to relatives, and in some cases people may even go further and opt for instance, PGT because it's a likely because they consider the variant as likely pathogenic or pathogenic. Next slide, please. So the PVS1 supporting was applied correctly by 89 participants, and the PM2 at supporting was applied by 142 participants. And the 77 who classified the variant as pathogenic or likely pathogenic, it is mainly due, as you can expect, by applying PVS1 at a too high level. So there were several people who applied it at very strong or moderate level, and they combined that with often with PM2 at a too high level. So according to moderate, and there were even a few who considered it as a strong or very strong argument, which has never been mentioned in the guidelines of this. So this is an example that it can be very important to really follow the guidelines. There are gene-specific guidelines for PALB2 published, indicating that especially truncating variants at the really end of the gene, there should be careful consideration to not misclassify these variants, because of course this can have a major clinical impact. And I think with this we are at the end of what I can tell for the PALB2 variant.
Great, Kathleen, thank you very much indeed, and for Arjun and Miranda as well. So just to summarise, it's always really nice to be able to have an overview of what classifications were submitted by all the participants. So here we have our bubble plot. So the larger the bubble is the more number of individuals who classified those variants, and anything in green is highlighted as the accepted or expected classification, and anything in red is not to be not considered to be correct. So here you can see we've got very good close correlation, particularly for variant 1 and variant 2, whereby we had 2 classifications depending on the guidelines applied that were accepted. Variant 4 as well, we've got very large green expected classification bubble there, which is nice to see. I think the most challenging one, I think, Kathleen, is the one that you've just discussed, whereby we've got more of a mixture across the boundaries of where clinical management may potentially change. So I think there's a bit of work to be done there on the non-BRCA1, BRCA2 variants. So next slide, please. So just to summarise, that we're currently assessing and putting together the data set for this Run 11 and include the feedback comments that we've just heard from our expert advisors, and also some questions that are coming in very rapidly on the Q&A function here, which is great, and we'll be able to summarise that in the summary report, which will be available early in 2024. Run 12, we're pleased to say, will be sponsored going forward as per this year, and we'll be able to provide this in spring next year, and obviously it will be advertised on the EQA provider website, and you'll be notified when registration is open. So next slide, please. And we are now going to open the question and answer part of the call. Simon, would you like to kick us off? I see, as I say, many questions coming in fast and furious, which is fantastic.
Yeah, we have, Sandy, which is fantastic. So just a reminder, if you are going to be submitting a question, please could you preface it with the variant you're interested in asking the question related to? But most, as I can see, have done that, which is great. Makes mine and Sandy's life much easier in terms of addressing it. I'm actually going to start off with a more general question, actually, because we as organisations, we put a lot of work into providing variants that sort of challenge the different spectrum of classifications here. And this, I think this one is really demonstrates that nicely. I've got a general point, probably, I guess, addressed more Miranda, I suppose, but in both cases 2 and 5, there is a difference in the variant classification depending on what guidelines you use, but it's really striking in case 2, where, you know, depending whether you use CanVig or Enigma, you can get a significantly different classification, so either class 3 or class 5, which could affect clinical management. Do you have a general point about the potential impact of the differences in classification depending on guidelines you use, Miranda?
Well, there's obviously the clinical impact of those guidelines. So what I would say is that the CanVig guidelines were kind of designed to be interim guidelines while we were waiting for the VSIP guidelines to be out. And obviously Enigma are the kind of ClinGen appointed experts. But there are also some, as we've noticed from other VSIP guidelines that have been released, and there are some quite significant differences by different healthcare systems across the world and how they manage genomic testing. So we would always in the CanVig group evaluate a VSIP guidance and determine whether it is kind of appropriate for our NHS patients and how we do our testing and how we select patients. And on the basis of that, we have already, in kind of conjunction with talking to those VSIP authors modify both the P10 and the TP53 guidelines as well, because in particularly the US, the kind of a predominant healthcare system, that those genes are included, and the same with the kind of HRR genes are included on that ACMG 79 list that is kind of tested for nearly every patient that comes through the door, so even somebody with a neurodevelopmental disorder, whereas in the UK we have a kind of quite strict testing, as I know they do in lots of other countries in Europe, and so our prior probability of that patient having a pathogenic variant in that gene, because we're very much selecting for that phenotype, is likely to be higher than it is in if you were just testing kind of everybody. So we are in the process of kind of going through the Enigma guidelines, and you know, have already adopted some bits. As I said, we've been waiting for a new version of our general ACGS guidelines to come out, and have been a little bit cognizant that we don't want to make too many changes when we know that then we're going to have next year the whole new ACMG guidelines come out. So it's a bit of a balance. But obviously, yeah, the CAMVIG guidelines have been established for a while. So yeah, now the Enigma guidelines, and also the Enigma have taken the decision to not include that kind of case control data. And at the time that was discussed, how we were doing the case control data at the time when it was discussed with Enigma, was when we were comparing it to Nomad. I think if now we've changed CAMVIG to look at UK Biobank, I think that's a much better, you know, population match control. So I think potentially, yeah, we've got kind of some further work to do to have further discussions and see whether Enigma would accept that kind of NHS versus UK Biobank data as a kind of an authorised case control analysis.
So I guess the sort of the take-home message is an element of caution required, and maybe a waiting game, if you like, until the new ACMG guidelines are actually published before we can really draw some, you know, make a strong decision about how we might, which way we might go with these classifications.
Yeah, I think the key message in all of this is, particularly for those involved with seeing patients, it's that genetic counselling, isn't it? And understanding that variant classification is not black or white, as I'm sure all the participants who well know,
Yeah.
that there are a lot of kind of nuances and complications, and it can be subject to change. And so that, you know, that's why a lightly pathogenic, it's 90% likelihood of being pathogenic. There's still that 10% that it might not be. And we've seen with other variants that come up that suddenly the evidence has changed, etc. So it's kind of us feeding back that, yeah, that information back to kind of, yeah, those clinical colleagues, and making sure that everybody's aware and that we have good guidelines in place for variant reclassification as well.
Yeah, and it just reinforces why it's so important to have good education and lots of experience in this area, just to make sure that, you know, we can impart that information clearly to the end user of the report of the service. I think probably in terms of questions, Sandy, maybe we should take it. We've got lots of questions coming in, obviously, that are now being classified by the different variants coming in. So if we start off with variant 1, which I think was you, Kathleen, I've noticed there are a couple of questions relating to understanding the difference between B70 and PVS0. Could you just reiterate? I know you described it in the slides as well, but could you just again just explain what the difference is between them and how it impacts that classification, please?
Yeah, well, for me the question was not completely clear. I understood that yeah, we apply the BP70, it's for observed, or the PVS10 for observed, if there is an if cDNA analysis has been done. And this, yeah, there is one publication where they have tested this variant. However, it's with the mini-gene assay, and they saw some leaky splicing. So that's why you can apply the observed code because it is observed. However, since it is a mini-gene assay and not real patient material, the strength, and there is only one assay published. The strength cannot be strong or very strong because there is only one assay. And then we realized that some labs may have contradictive results obtained in patients, so that the mini gene assay was not reflecting what is actually seen in patients where there is no aberrant splicing observed.
Fantastic.
I saw there was also somebody who was a bit confused about the fact that if we could apply BP4, and because we discussed there the cutoffs for BP4. It should be less than 0.1, and predicted by Splice AI, and Splice AI predicts a score of 0.04, which is indeed lower than the cutoff proposed. However, if you check the guidelines for ATM, it is clearly indicated that BP4 may not be applied for splice predictions in conjunction with BP7 observed a lack of observed RNA defect. And this is, I understand that it is confusing, because in the CANVIC guidelines it is indicated that for silent variants you can combine BP4 and BP7. However, specifically for if there are cDNA analysis available, you cannot combine BP7 observed with the prediction BP4 for splice prediction. I hope this makes it a bit more clear for
Thank you, Kathleen, because that was my next question. So you sorted it. You've done. So thank you. Sandy, should we go on to case 2, variant 2?
Yeah, can do. Actually, I was going to jump to variant 3 because there's quite a clear question here, and I think, Miranda, that was one of yours. So it's the BRCA2 variant. So since it's an in-frame deletion, should we use PM4 instead?
No, so you'd use PM4 for a small in-frame deletion, so a few kind of amino acids, because this is a copy number variant that's involving one or more exons. Then you would follow the PVS1 decision tree and use it kind of through that route.
Yeah, thank you.
Downgrade the strength, yeah.
Yeah, no, that's great, thank you. And then another one for the same variant is, so if the variant's not in NOMAD, should not be used PM2 with moderate strength as suggested by CANVIC, or it was present in the UK Biobank?
Okay, so we can't check the UK Biobank data for these structural variations, and it's quite easy to get hold of the WGS portion of UK Biobank, but not the WGS portion. The AC and GCMV guidelines by Riggs-Rooney et al. Do not have any kind of points towards pathogenicity for rarity in controls. And we have had numerous kind of toings and froing from the ACGS with those authors and trying to decide whether we can use anything to make, because at the moment with the CMV guidelines, you could use something like a large deletion, a whole genome gene deletion as a standalone piece of evidence. In the SMV guidelines, you can't. So even if you could apply, say, for example, PVS1 very strong, you would still, if you didn't have any other evidence, you'd still have to classify that variant as VUS. So it's a real discrepancy. So we have had agreement from them that particularly with the NOMAD version 4 and those 70,000 genomes, there is sufficient data, and way more than we had originally in the EXAC kind of population. We first started using PM2 to use PM2 at a maximum strength of supporting. So that's kind of what we have been advising in the UK. I think, yeah, moderate is potentially a bit too, yeah, you're overweighting that rarity data.
Great, thank you. That's really, really important advice. Thank you for that. And I've got one final one. I'm going to stick with the same variant. So in Enigma, why PP1 is not applied?
So it sort of is. Yeah, there are some papers that talk about PP1, but actually if you look at the Parsons et al. Multifactorial data, as I said, it was split up into kind of the different sections they use. They look at segregation, and actually the majority of that likelihood ratio came from the segregation data. So we'd kind of already included it in that multifactorial. There is also a slightly argument we could have found other papers potentially that weren't included in that Parsons et al. Analysis. But once you've got a classification of class 5 and you've got such a kind of reported variant, then you're just kind of almost making a bit of extra work for yourself. But yeah, that PP1 was kind of really in that multi-pack.
Yeah. So again, going back to the important point that you can't use the same evidence twice. And be
not counting the same families.
Yeah, be aware of that. Absolutely. Okay, Simon, do you want to?
Yep.
Another variant.
I've got one coming for you, Arjun. So it's variant 5. So there are a couple of questions for variant 5. The first one is, why not use BS1 supporting when there's a nomad frequency of 0.003% in a non-finished population?
That's a good question. But for we have to use the filtered score, and that is I think we've answered that also in a nomad. You have to use the PopMap filtered score, and that is too high to apply them for the PP1 support. I can't do it by heart, but that score is slightly higher for this variant, and then you take into account the standard deviation around the frequency, and then that score is still too high. But I can imagine that you would use this score, but because you have to use the FAF score, you can't apply it.
Okay, fine. So I mean, that leads on to the next question, which is really around BS3 and why not using PS3 rather than BS3? So if the defect's less than 10%, why are we applying BS3 instead of PS3?
The S3, you should apply that when you expect a deleterious effect, and we do not see any evidence of a deleterious effect. So that's why you shouldn't use a score towards pathogenicity, because there is hardly any effect at all.
Yeah, perfect. Thank you. Sandy, over to you.
Yeah, I've got one here that's sort of more general. So can you please explain how you use PS4 and PP4 together, as there's a caution in the combination table that we just presented? So the BRCA2 SNV and the BRCA2 exon 14/16 deletion. I don't know who wants to take those ones. Miranda, those your ones. One of them is, isn't it?
Yeah. So the for the BRCA2 exon deletion, then we're obviously using the Enigma multifactorial data, but that's not including the UK data. So then we did the PS4 based on the kind of that incidence of 7 counts within our UK database, and then the Rebecca et al. Score, and adding that at a moderate. But yeah, that's that difference between the case counting approach allowed by CAMVIG and not by Enigma.
Okay, great. Thank you very much for that clarification. Where do we want to go now? I think there's a lot of chat being submitted about how frustrating and confusing it is to have guidelines that don't align. So hopefully, we've talked about how some of them are interim and we're waiting for clearer guidance coming out. And also somebody's very helpfully popped in the chat about there's a ACMG AMP CAP ClinGen webinar, which I believe is today, on the standards for sequence variant classification version 4, the draft, and Steve Harrison and Heidi Ream are going to be providing a summary and presenting that. So perhaps you may want to Google or any other web browser to find out more details to join that if you're interested. So thank you for sharing that.
And that specific webinar is also available on the YouTube channel already from ClinGen, which is very informative to follow to learn more about the version 4 of the ACMG guidelines.
And that's brilliant. In fact, we'll maybe put that link in our EQA summary report for those can reference it at a later date. That would be incredibly helpful. Thank you.
Yeah, but for now, it will only lead to more confusion, of course, because in the end, we have to see how these guidelines will be translated into the gene-specific guidelines, and it will take us at least a year before we have that information again. So
absolutely,
yeah, I'm afraid it will take quite some time before it will get easy.
Yes, and I think they're in the process of being applied, aren't they, to lots of case scenarios, etc., and trying them out. So yes, we'll see, wait for the publication coming out. Simon, over to
Sandy,
you.
I think we've got quite a lot of just sort of very specific targeted questions relating to the particular variants, which I think would be probably best to be picked up in the scheme report. I think we've got one more last question, which is a more general question, I think. Again, I think it's going to come towards you, Miranda, because you mentioned this quite a lot. Can you explain the general principle of leakiness a bit more, please?
Yeah, it's not a great word, is it? You could say kind of incomplete splicing, but it's well known. People say it's leaky splicing. So we know that different genes have different thresholds for how much of that kind of wild type protein is required for normal function. As I said, for BRCA1 and 2, then Enigma have set that cutoff as 80%. So you have to have at least 80% or more abnormal transcripts in order to kind of use that RNA evidence towards pathogenicity. So you just, in the case that I presented, there was 85% of the transcripts were abnormal. So we are, relatively confident that that is then below that kind of threshold where you are going to see that effect, in the biology, and then kind of produce a risk of cancer. But as I said, if you have got assays, particularly where you've got multiple assays showing some of the leakiness, and the leakiness just means you get some normal transcript, some full-length wild-type transcript produced, then you need to be quite cautious about your application of that data. And in this case, we were very lucky. We had 2 good assays that both kind of quantified it about the same level at 85, and so we were okay to still apply PS4, but we downgraded the evidence, and that was both in the CAMVIG and the Enigma guidelines. For older assays where there isn't kind of that same quantification, then again I think it's using your scientific judgment and being cautious, and again, if you've only got one assay and there isn't quantification, then you really want to be thinking about what the other evidence for that variant is. Do you really need to use that evidence? If you're just using that evidence to get it over the line, it's trying to kind of seek other evidence to make sure that you're kind of confident with that classification.
Thank you, Miranda. That was a really nice explanation of it. Hopefully that's addressed the question from the Q&A.
Perhaps can I add the Walker et al. Paper, which has been published in July in the American Journal of Human Genetics, on how to apply splicing variants, how to use splicing information into variant classification in general, and what percentage of leakiness can be converted. And of course, it depends a lot on the gene you're interested in. And we know for BRCA1, for example, and BRCA2 from some examples, that about 20 to 30% of wild-type transcript is enough to have a functional HRR function. So yeah, that perhaps is
yeah, and I will just want to add, because I have also used the word leakiness during the discussion of the variant. And to my knowledge, the cutoff is not yet defined on how much you really need for ATM.
Thank you all, because I think it's really nice to sort of clarify that. And of course, we will get a summary of that into the summary report anyway. So we'll clarify that again, and you'll have that information available in the summary report. Sandy, should we call it a day on the old questions and answer front there?
I think so. I think we've worked our expert panel very hard today. So a huge thank you from all of us, both in the EQA providers, but also our participants, who you really do help make things much clearer.
Fantastic. Right, well, we'll wrap it up then if that's the case. So just to reiterate what Sandy says, it's a big thank you to you for dialing in. We hope you found this webinar useful. And of course, obviously, we'll make it available on the EQA providers website, so you can listen to it at your leisure when you need to. A big thank you to our speakers, because not only have they done a fantastic job today, but also they're the people who've made this EQA scheme really, really work. So thank you to you for your support and helping make it so good. A thank you to our to my MSD and AZ for this non-promotional webinar. Without them, obviously, wouldn't get that out to you. And finally, a big thank you to my co-host Sandy as well for all her work. And we'll look forward to catching up with you in spring, I think, next year with Run 12. Do keep an eye out on both providers' websites, but also an eye out from the email, which will announce the next run when the run will be open to you. So thank you once again and have a great afternoon. Thanks for that. Bye.
Thank you, everyone. Bye-bye.